Surrounding the entire muscle is the epimysium. The whole muscle is made up of many fascicles. Every fascicle is encircled by the perimysium. Both the epimysium and perimysium are dense, irregular connective tissue. A fascicle is a bundle of 10 to 100 or more myofibers. Each myofiber has little thread-like structures that extend across the entire fibril, called myofibrils. Their prominent striations make the entire skeletal muscle appear striated.
A nerve action potential from the central nervous system arrives at the axon terminal, and this causes the calcium channels of the membrane to open up, allowing calcium to move into the synaptic knob, which causes the synaptic vesicles filled with acetylcholine to fuse with the membrane and release the acetylcholine into the synaptic cleft. The acetylcholine will then diffuse across the synaptic cleft to the motor end plate, where two molecules of acetylcholine will bind to an acetylcholine receptor, causing a sodium-potassium channel to open. Sodium will then travel through the channel to the muscle fiber, making it more positively charged, causing a muscle action potential. Once the nerve action potential stops, the leftover acetylcholine will be broken down into acetic acid and choline, which cannot activate the acetylcholine receptors. They are broken down by an enzyme called acetylcholinesterase. This action potential will travel along the sarcolemma into the T-tubules and will trigger the release of calcium from the terminal cisternae of the sarcoplasmic reticulum, where it was stored in the calsequestrin into the sarcoplasma of the muscle cell. The calcium from the sarcoplasmic reticulum floods over the thin filaments, beginning the process of contraction by binding to troponin, which pulls the tropomyosin over and exposes the active sites known as actin. Meanwhile, ATP is produced by a series of oxygen-required reactions in the mitochondria. The ATP binds to the myosin head and is hydrolyzed for energy, leaving ADP and a phosphate. At this point, the myosin head goes into high energy state and it leaps over to the exposed active site, forming a cross bridge while the ADP is still attached. The ADP is then released, and then the myosin head slides the thin filaments past the thick filaments. This is called the power stroke. After the power stroke is completed, the cross bridge will stay until the myosin head binds to another molecule of ATP, causing it to detach from the actin active site and go back to a low energy state. The calcium will then be pumped back into the sarcoplasmic reticulum where the calsequestrin is. This will allow the tropomyosin to cover the actin sites again so the muscle may relax.
Go.
Basic functional units of a myofibril are compartments called sarcomeres. The components of a sarcomere include the Z-discs, which are narrow plate-shaped regions of dense material that separate one sarcomere from the next. A Z-disc passes through each I-band, which is the lighter area that contains only thin filaments and is also less dense. The A-band is a dark middle area of the sarcomere that extends the entire length of the thick filaments. It also includes the part where the thin filaments overlap the thick filaments. The H-band is the small area of the A-band. That's the end.